This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 592, recorded on March 18th, 2020. I'm Vincent Raccaniello, and you're listening to the podcast all about viruses. Joining me today from Austin, Texas, Rich Condit.
Hi, everybody. How you doing? I don't even know what the weather is here. It's cloudy and, I don't know, mid-70s, something like that.
Cloudy and pandemic.
Cloudy and pandemic. Cloudy with a chance of coronavirus.
Also joining us from Western Massachusetts, Alan Dove.
Good to be here. It's partly cloudy, 52 Fahrenheit, 11 C, although I have not been outdoors yet today because I've been grinding through all these letters we got at TWiV. But maybe later today I'll walk around, in a socially distant way.
And joining us here in New York City, Ori Lieberman.
Hi, guys.
Welcome back. You were just here the other day.
Yeah.
Recording a special TWiV. And Ori, of course, is from This Week in Neuroscience, twin. And he is an MD PhD student here who has had his rotations canceled. So you have a little time on your hands, right?
Yeah, a lot of time.
So I said, come over to TWiV.
Hey, Vincent, a question just occurred to me.
Yeah.
Because you are in the city. You're at Columbia, right? How was your commute?
No, this week has been non-commute. I mean, it's just there are no cars. I just breezed right through.
Is it spring break?
It is spring break, but
okay.
I think next week it's going to be the same, most likely. The only thing is that there's still a lot of trucks on the road. So on the George Washington Bridge, the trucks have to take the upper level, and that still has backed up from truck traffic, all trucks. So there's some kind of commerce still going on. But the lower level, which is cars only, breeze right through. It's nice. Now I understand I probably shouldn't be coming in, but we're trying to make a plan to shut off our lab this week. We've all been told to ramp down to essential activities and accept people doing coronavirus research. So we're actually starting to do some coronavirus research, but with Daniel Griffin because we think it's important. And in the event that the city shuts down, which I think is unlikely, but in the event that it does, we have to label which cages of mice we no longer want. As if there was ever a cage of mice you didn't want, right?
Wow.
Yeah, well, many people are impacted, right? That's the way it is in a pandemic. Yeah, so this is a special Wednesday TWiV. It is the fourth. Let's see, we had Sunday, Tuesday, Wednesday. So this is the fourth for the week. We'll have one more because people have asked for more. We have a lot of email. And so thank you, everyone, for joining us. And we'll try and get through some of this. We have a few interesting quick stories to begin with and then some email. If you like what we do, consider supporting us. Microbe.tv/contribute. Where else can you get factual information about viruses? We've been doing this for 10 years, and it's free. So we only ask for your help. A couple of things that came out in the last 24 hours. This is what everyone's talking about. This Richard wrote in and sent it to us, but many people have, you call it scary modeling. It's a paper out of Imperial College, COVID-19 response team, impact of non-pharmaceutical interventions to reduce COVID-19 mortality and healthcare demands. Who has a good summary of this? Does anyone? I could do it, but I just wondered.
I don't.
All right. So basically they are saying, unless you lock down, close schools, universities, keep people at home, it's going to be bad. And the healthcare system is not going to be able to handle it, which is something we've known for a while, right?
And they're saying if you do all those things, it's going to be bad, but less bad.
Yeah, but the problem, this is, of course, a model, right? And as we know, all models
are wrong.
Some
are useful.
All models are wrong, or most models are wrong. Some are useful, something like that. So I don't know how accurate. All I do is I tell people, look, in China, they locked down for 2 months and they could handle it. And so there's no reason why anyone else could do that. Look,
China would like to believe that.
Let's not look behind the scenes. I know there are lots of ifs, ands, or buts. on the surface, it looks like, so there's nothing you can do except either emulate or do less. We're not going to emulate them. We'll do less.
No.
What are you going to do? I don't see what the point of doing these modeling is. What are they trying to scare policymakers into doing something?
Yes, essentially. Now, what's going on in the UK, I think a little context is important here, is Boris Johnson, the Prime Minister of the UK, initially was advocating a really kind of a laissez-faire policy. Rationale was eventually we're going to have to get enough people to get this thing that will have herd immunity, and so we might as well just get that out of the way. And it's the chickenpox party model, like I call it, where back in the old days, before there was a vaccine for chickenpox, sometimes parents would get their kids together to spread it deliberately so they'd get it as kids, so they wouldn't, they'd be a little less likely to get complications. So Johnson initially was kind of advocating that type of approach, and then public health people pointed out, that's going to kill many thousands of additional people beyond what would otherwise be dead if you kind of mitigated it. And so I think this modeling group, which is at Imperial College London, is working in that context and trying to project what exactly would that entail. And so that's where we get these curves.
So they write here.
That's not to say these curves are wrong either.
We don't know. It's a prediction. It's a model. It's just based on what we know. So they say major challenge of suppression is that this type of intensive intervention package or something equivalent will be needed to be maintained until a vaccine becomes available, potentially 18 months or more, given that we predict that transmission will quickly rebound if interventions are relaxed. Okay? So that's why China is an important model. They are beginning to relax interventions. Let's see what happens there. And if they do propose,
they propose in it, there's this where you just lock somebody up for 18 months, which of course isn't going to happen. But they also have a somewhat more realistic model where they look at if we finally get around to making tests widely available, then could have an adaptive triggering mechanism where we're going to lock everything down for a month and then we'll hopefully get cases under control and ICU beds back to functional level. Then we're going to start letting people go out and we'll keep testing anybody who gets sick. And when cases start going up again, then we'll lock everything down again.
So the point here is that we need a pandemic policy, which we don't have. Every country needs one. It's hard to do this on the fly. And that's what we're doing anyway. Some countries started in January, but I hope people in the U.S. at least realize that next time we need a pandemic plan in place. And there are lots of parts to that which we don't need to go into, but we don't have one. And that's part of the trouble.
You guys have been discussing what for me is the elephant in the room, and the question that my wife is asking all the time, which is, okay, what happens when we relieve the restrictions?
Yeah, we'll see in China.
I think the answer is we don't know.
Don't know.
Okay.
Don't know.
So I heard about this thing called the pandemic yo-yo, where we would have 4 weeks of restrictions. We'd get out for 2 weeks or 3 weeks, and then we'd all go back for a couple of weeks, and that would at least keep the economic burden from being too high.
That sounds to me like the old policy of giving an antiviral for a while and then withdrawing it. That was the worst to get resistance, right? Yeah. I think you have to bite it for two months probably, right? Which is, I mean, there are some people, and Steve Morse mentioned this yesterday, who can't handle being inside for long periods of time without social interaction, right?
Are you guys worried about in a week, if it doesn't get bad, people saying, well, it's not that bad, so we go back outside?
Yeah, sure.
Yeah. I think that's basically what happened is that especially people who feel themselves at low risk are going to disregard this very soon, start going about their business, and we may see a rising case counts at that point.
Mm-hmm. Well, that's where stuff like actually legislatively shutting down bars and restaurants and that kind of stuff makes a difference.
Mm-hmm. I think here in New York, everything is shut, right?
So it's funny. First of all, yesterday I went to the grocery store and things had completely restocked from the weekend, but everything was still open. The nail salons, the hair, like the barber shops. they were empty, but there were people inside.
But restaurants are closed.
Restaurants are closed except for takeout and pickup. Yeah.
Yeah. Okay. All right. So we have another story about this drug, Avigan, right? Which I was looking at last night. Did you put that in, Ori, or was that me last night?
No, I think that was you.
So Avigan is an interesting. It's an antiviral against influenza, which is licensed in Japan and perhaps some other countries for influenza, but there's some evidence that it might be working against coronavirus.
What is it?
It's favipiravir.
It's a polymerase inhibitor, right?
But the data is not released.
The data
is not released. Yeah.
What sort of polymerase inhibitor? Is it a nucleoside analog or a non-nucleoside inhibitor?
Let's look. Favipiravir. I should know this. It looks like a nuke to me. There you go. 2 rings. Yeah, it's a nuke. It's a nuke.
Okay.
Nucleotide analog.
And it's all called T705. I was just revising the antivirals chapter for the textbook, and I was reading about it, and it said the U.S. has no intention of licensing this for influenza in the U.S., even though it's licensed elsewhere. Anyway, we'll see if that works. We actually have some other leads on antivirals we'll talk about. Actually, let's do the antivirals now since we're on it.
Sure.
So just quickly, the interesting patent thing with this drug is that it's patented in Japan, but off patent in China. So they're actually making generic. So it's a Fujifilm-owned scaffold that is now essentially being made freely in China because the patent expired.
Interesting.
Yeah.
It's interesting that it just goes to show that you might as well test all the approved drugs, right? Because you would not know it's the RNA polymerase of influenza virus. Really, it's going to inhibit this one. Well,
there are. I'm sure companies are doing this already. There are panels of every approved drug.
Yeah.
And you can just screen them against SARS-CoV-2, see what's active, and they're all approved drugs. So start there.
Yep. Another drug just sent to me this morning by a journalist. It's a paper shared from a French group in Marseille. And these are people who we know from giant virus, the world. Didier Raoult, Philippe Colson, Bernard Lascola, and many others. But hydroxychloroquine and azithromycin as a treatment of COVID-19. They did an open-label, non-randomized clinical trial. Open label, of course, meaning it wasn't blinded. It took 22 patients with laboratory-confirmed infection, ranging from asymptomatic, mild to more severe, lower tract infections. And they treated them with a regimen of hydroxychloroquine and azithromycin. And they reduced their virus loads, and clinical symptoms were also reduced. And chloroquine, of course, would inhibit virus entry, would prevent the acidification of endosomes, well known to inhibit many viruses. And so, as many people may know, chloroquine is used in some cases for malaria treatment, right? Or maybe it used to be used.
It's still used.
Yeah. Is it still used? It gives you weird dreams, I understand. But hydroxychloroquine, apparently less side effects, but not as available. So that's a possibility.
And azithromycin?
Oh, that's an interesting. So that's an antimicrobial, which has for many years. I asked Daniel Griffin this today. He said it's also, randomly found to have some antiviral activities, because in their trial actually it worked better with the addition of azithromycin, which I think also has anti-inflammatory properties. So maybe that's part of it.
So, and hydroxychloroquine is used in rheumatologic diseases as an anti-inflammatory as well. So it's under the trade name Plaquenil.
And so this isn't quite as random a choice as it sounds.
No, and the other thing that's interesting about this is that, we'll talk about clinical trials in a second, but there's a trial in Minnesota that is testing it as a tool for post-exposure prophylaxis. The idea being that if you prevent viral entry very early, then you actually have a stronger effect of the drug because you would get it right after exposure. And they're actually enrolling and will send you the drug from like it's not just localized to Minnesota. So interesting.
So if I get too close to somebody who might be infected, maybe a gin and tonic is in order.
So this raises the question of whether healthcare workers could be given this prophylactically.
Yeah.
Does that make sense, Ori? You think?
Yeah, everything has to be based in data.
Yeah.
So
I like that. Everything has to be based in data. You mean Twitter is not good enough?
Well, there's...
I'm just kidding. Just kidding. Anyway, so this will be... Many people will be doing this trial because these drugs are available, right? Mm-hmm. They're licensed. So we'll see more on that. Then I wanted to mention the the RNA vaccine that's just gone into phase 1 by Moderna, or Mode RNA, whatever you want to call it, because that's the original name of the company, Mode RNA, which it's an RNA-based vaccine. They make RNA and they inject it into you, so it's coated with protective lipid nanoparticles, and then it's injected. And this small company is basing its fortunes on these mRNA vaccines. The idea is the mRNA is injected into your muscle. It makes protein, it's translated into protein. The protein is immunogenic, and you get an immune response. And they have a number of different viruses they're trying this for. Back in January, they got the sequence of the spike. They made an mRNA. They put it in mice, showed it was immunogenic. And now they're in a phase 1 together with NIH. So this is cool because it's a very rapid platform where you don't have to make virus, right? You don't have to deal with virus production. You don't have to make vectors and so forth. And you could do it in principle very quickly. So if you go to clinicaltrials.gov, you can see the phase 1, 45 patients for safety. They're going to do several doses. And then the primary goal is the primary objective to evaluate safety and reactogenicity. 2 doses given 28 days apart. And then the secondary will be to measure immunogenicity. They'll measure antibodies against the viral spike glycoprotein. And this is how many days they're going to do this. I'm not
12 up to 12 months.
12 months observation, but the initial data on which they will base subsequent trials, phase 2 and 3, should be quicker than that. Anyway, it's going to be rapid. They're saying by the end of April, they'll have the data, whether it's safe. What do we think? Do you think it's going to work? It's going to be protective?
Who knows? I mean, these things, who knows? I mean, it's like, it's interesting to think about this as opposed to other vaccine strategies, because it's similar in a way to just to a subunit vaccine where you're just administering a protein, right? Yeah. But it's different than that, it seems to me, in several ways. First of all, I wonder if and I need to learn more about this, but these are mostly questions at this point. I wonder if by using a messenger RNA, you get in effect, a dosage amplification, because each messenger RNA molecule is going to be translated a number of times, number one. Number two, one of the big problems with subunit vaccines is getting the right conformation of the antigen. And since this is made in the cells, I think there's a better chance of getting it folded and presented properly. Okay. Number three, since it's being processed in cells, I would think that there would be a better chance of getting both an antibody response to the full protein and a cellular response to peptides being presented on the surface of the cells expressing the antigen. Okay. So all of those things combined may actually confer numerous advantages over just a protein vaccine. I don't see any reason why it shouldn't work, and certainly it appears to be working in their animal models. But beyond that, vaccines are kind of black magic. You don't know necessarily that you got the right antigen. No, exactly. Or we don't know for sure what the correlates of protection are, I would assume, okay? And, quite often you miss the bus with these. So it's still kind of a trial and error sort of thing. The other thing that occurs to me is that the safety considerations with this, it seems to me, are entirely different than many other vaccines. Okay, because you're not dealing with you're not dealing with a virus. You're not dealing either with an attenuated virus or a wild type virus that you've inactivated. It is much more like a subunit vaccine. So the safety considerations, I think, I can't. It's hard for me to dream up how this would be harmful.
Well, it was hard for people to dream up how an adenovirus gene therapy vector would be harmful, and then somebody got killed that way, and then gene therapy got shut down for years. So, but you're of course correct that the safety issues here are different. That doesn't mean that they're non-existent, but it certainly looks promising.
Are there examples of this technology used in an existing vaccine? That's what I'm trying to look for over here. But
I don't think so.
No, this is new.
No, I think they were developing this platform for a few things, and since they had the platform, and obviously since it's RNA, once you have the sequence, you just make it.
I see.
I mean, there are DNA-based vaccines, right? In horses, there's a West
Sure.
Nile DNA-based vaccine, and but in people, they haven't worked all that well. So this one is interesting because not only you can get the sequence quickly, you can optimize it, you can do codon optimization, you can pick the exact part of the protein you want, and so forth. So it's very interesting, and we'll see if this works, and then if it can be extended. I mean, it's a great platform for a pandemic where you have all of a sudden a new virus, and you don't have anything. So the next flu pandemic, if this works, you could do the same thing.
That's the big question, if this works. So it's a great platform because it's really quick, but if it's really quick and doesn't work, then that...
Another subtlety of this is that it's the pre-fusion stabilized spike protein. So during the process of entry, the spike protein goes through dramatic conformational changes. And it's thought, I presume, that the conformation that exists on the particle before fusion is the most antigenically protective conformation. And so they've tweaked the sequence based on good old-fashioned basic science in a fashion that should stabilize the protein to prefer its expression as the pre-fusion conformation, and then thus enhance the protective effect.
Right.
So we'll see.
We have the paper we talked about last time on the stability of SARS-CoV-2 and aerosols and various surfaces. That's been published in New England Journal, and that's making a big splash today, although it's been out in medRxiv for some time. Same conclusions. But in the same issue, I found a couple of interesting papers. New England Journal of Medicine. I don't know if issue is the right word anymore. March 12th. Who knows? Detection of COVID-19 in children. All right, so that's not right, unless you're talking about disease.
Right.
Maybe it is disease. Detection of COVID-19
SARS-CoV-2
in children in early January 2020. And this report is basically a study of 366 hospitalized children less than 16 years of age. And the key here is that some of the younger ones did, in fact, have lower respiratory disease, because we've been saying, kids aren't getting sick. But
well, actually, so detection of COVID-19 is correct.
Yeah, it's disease.
They did detect disease.
Disease in children causing moderate to severe respiratory illness in the early phase of the Wuhan outbreak, associated with one ICU admission and one patient. So it's rare still in kids, right?
Yeah.
It's just, the point is, when we say it doesn't make kids sick, we probably shouldn't say that. It makes them sick less frequently.
This is right. And this is an important point, because a lot of people are going around, I'm 25 years old, I'm not at risk. Well, nobody is not at risk. There's no zero risk group that we can find. And so, even kids potentially, yes, the risk goes up dramatically when you get above about age 50, but the risk is not zero for any age group.
I should say that, to clarify, that this was a study of all respiratory infections in these 366 kids. And among them, they found influenza A in 23, influenza B in 20, and SARS-CoV-2 in 6. And it was one of those 6 that had to be hospitalized.
Right.
So that's cool. And all these things you can access. And
then there's a paper in the journal Pediatrics that goes through, I think, 2,500 kids in China. And they pull out, young age is obviously a risk factor of worse disease. I'm sorry, I'm trying to find the list of other things.
This is COVID-19 in children, initial characterization of pediatric disease, right?
Yes.
Yeah.
And then there's a second epidemiological
younger age,
characterization.
under pulmonary pathology, and immunocompromising conditions.
Exactly.
So again, we don't have zero disease in kids, right?
Yeah. And especially in populations of sick kids who have these very severe underlying diseases. Yeah.
Yes. Well, children are less likely to become severely ill than older adults. There are subpopulations of children with an increased risk for more significant illness. Right. That's a good point. Okay. And a few more things here of interest. You have 2 papers. Is it the same thing? Yeah, I think it looks like this.
Yeah.
Oh, no, it's 2 different papers.
These pediatrics publications are duplicates.
Sorry, yeah.
Yeah.
Another, in the same issue of New England Journal, SARS-CoV-2 infection among travelers returning from Wuhan. These are patients that were evacuated, 94 persons evacuated from Wuhan to Singapore on January 30th. And they developed infection in the course of the flight, and afterwards they could monitor them because they were kept together. And it's clear evidence for asymptomatic transmission, I think. So a lot of people are saying, no, there isn't any. It doesn't contribute. Yes, Ralph last week said clearly there's asymptomatic transmission, but here's some good epidemiological evidence that there is, in fact, asymptomatic transmission. So a nice figure of the follow-up after the flight and so forth. So that's worth looking at. I think you can get that because I got it from home this morning.
These are all, so far these have all been accessible.
Another article.
I put this one in the Science paper.
A modeling of asymptomatic infection.
Ryu and Lee et al.
Oh, yeah, that's from our colleague here, Jeff Shaman. Substantial undocumented infection facilitates rapid dissemination. So that's, again, like asymptomatic or mildly symptomatic, right?
Mm-hmm.
Infection. And this is a modeling, again, as well, right, based on existing data.
Mm-hmm.
We estimate 86% of all infections were undocumented prior to 23rd January. So maybe we should have Jeff on to explain this. But in the bottom line is there is a contribution to transmission of undocumented infection, right?
Yeah.
It's a good paper. And then there is new things keep getting inserted.
Right. So I put in the latest, the CDC, this just came out in MMWR as I was preparing for this episode. After I'd gone through the emails, I went and looked online in my news feeds. So Morbidity Mortality Weekly Report just has this paper out for early release, Severe Outcomes Among Patients with Coronavirus Disease 2019, U.S., February 12th to March 16th. And these are the U.S. data on case fatality rates. Age group over 85, fatality rate ranged from 10 to 27%. Then it was between 3 to 11% for ages 65 to 84 and 1 to 3% for 55 to 64, and then less than 1%, 20 to 54.
I'm glad they stratified by age.
Yes.
Right.
Now,
really
the big caveat on this, since it's a U.S.-based study, is that our testing situation in the U.S. has been, it's a family show, so I'm trying to figure out the right term to use for this, but a train wreck. It's been very, very poor, and we've done very little testing, and we really have no frickin' idea who's infected because of it. So these are patients who, for the most part, showed up at the hospital and happened to get tested. That tiny minority has contributed to this report, but the numbers are kind of in line with what we're seeing elsewhere in the world.
So they could be much lower because we probably have many
we
more
probably
infections.
haven't, as we were just talking about asymptomatic and minimally symptomatic, none of those people are getting tested.
Yeah.
But I don't know if this has been covered yet on earlier episodes, but there's finally now that the FDA has opened up the possibility of commercially developed tests, Roche has developed a test kit that is shipping out already, and that's going to ramp up testing substantially. They're talking about already having 400,000 tests in stock and producing 1 million a month or some bigger, the kind of thing industry can do. So that should help a lot.
I'll just add two quick things. So New York Presbyterian has a morning teleconference. So this morning they actually said that now they have a much higher testing capacity in the lab, and they're out of nasal swabs. So before they were swabbing the nose and the
new
mouth.
Bottleneck.
Yeah. So now they're only swabbing the nose, and then they can double the number of swabs they have for patients. And then the other thing that was interesting was that it's actually, so regardless of the percentage of people who are hospitalized, the absolute number is really the problem in terms of the surge. And as of today, they now have 200 inpatients that are COVID positive. There was 120 yesterday. And by back of the envelope math, that's 10% of the hospital beds in the New York Presbyterian system. So it's ramping up very rapidly.
You don't know what fraction are ICU beds, do you?
So the rumor, this was not what they said, was that essentially half of the ICU beds are with COVID patients right now. So
Ori, I have a question that comes through my wife to a large extent. modified by me. First of all, what is the role of the hospital in this? Because up to a certain point, there's nothing that can be done, right? But at a certain point in illness, the hospital's going to help you out. Ultimately, to me, that translates, or at least what I've heard, is a respirator. So the question is, at what stage in this whole thing, at what stage of criticality can the hospital really help out? And what's a respirator? And what are the limits on their availability?
So we talked a little bit about this in the last episode, and I'd just say that, I guess hospitalization is helpful when you're having respiratory distress, and there are other types of oxygen delivery systems beyond respirators that can be given in the hospitals, like a nasal cannula or a mask that gives positive pressure without being intubated.
Okay.
And then you have all sorts of other type of supportive therapies. So you could imagine getting IV fluids to make sure that you avoid a shock, like a shocky presentation. And essentially, so going into so the triage point is whether they feel like you can make it at home by drinking water, managing your fever, and letting your body take the course, or whether you need to have support, either oxygen or fluids.
Okay.
Yeah.
And a respirator involves, is that intubation?
So you can be on a respirator with a mask that is, yes, essentially in this case, you will be intubated. And there's a lot of stories coming out saying that they're actually intubating much earlier than they would normally because it lowers risk for aerosolization later in the course of disease when the viral titer is higher.
Mm-hmm.
So essentially patients are getting intubated, they're put on respirator or ventilators, and then that hopefully gets them through the worst of it.
Someone asked me today if people who are intubated never survive. That's not true, right?
No.
You have a fraction who you can save that way, right?
Well, the whole point of intubation is to get them through, it's a backstop essentially to get them through the worst of the illness. Yeah. And that's what's happening in Italy. That's so scary is they're choosing who to intubate based on who they feel like can survive. Mm. So you could imagine that those are hard decisions to make and will be a problem. Yeah.
Yeah. Because once you're intubated, you have to be on a respirator, right?
Correct. Yes.
So they're deciding, okay, we have X respirators and X plus whatever patients, and who gets intubated, who gets put on a respirator.
Exactly. And you actually legally in the New York State cannot be removed from a ventilator to give that ventilator to someone else. So the choice will have to be made prior to the intubation.
Right.
Someone said on Facebook that they didn't care what a medical student's view of this was, but, I told them go away, but obviously you know way more than we do. So.
And I don't know that much. So, but I tried to recruit a resident here who is going into critical care, and he said that he's too busy. He can't get out of the hospital.
Understood.
Yeah.
Understood. So speaking of shortages, I went to get Tylenol. There's none left in the drugstore I went to. Of all the different sorts of Tylenol, right? Every flavor. And I went to buy a webcam at Staples, and they have none left because everyone's working at home.
Gosh, right.
And, I'm kind of disappointed in our capitalist system that can't live up to a demand like this. Isn't it crazy? I'm really disappointed in it.
And there are weird shortages too. Yesterday, I've been doing just my regular grocery shop, so yesterday was Tuesday. I went to the store, and of course the toilet paper is still in stock, but then I wanted to pick up some chocolate chips to make cookies, and they're sold out.
Wow.
Everybody's at home making cookies, I guess, is the thing. So
I think all this can get restocked, but it's kind of
course it will. I mean, everybody panicked and hoarded weirdest stuff. But
my daughter works at Trader Joe's, and she said yesterday that the truck comes in at 5 a.m. and they all work. And she said, we have everything. We don't have toilet paper. I don't think they even sell it there. But she said, we do have wipes. All right. Two more papers. One, this is not a paper. It's in the Guardian, but there's a report out of China that the first COVID cases were in November of last year. They have a cluster of 266 people who seem to be with serious respiratory illness. And I don't know that we have any diagnosis of them, but this would suggest that it was circulating even earlier than we learned about. which is not surprising to me at all. It was probably circulating undetected. Did you know, by the way, we dealt with this earlier on TWiV. Many people blamed a lab in China, and now that China's blaming it on us. Isn't this crazy? They say it came out of the U.S. Oh my gosh, this is just terrible. One more paper just was sent to me this morning, bioarchive from a group in somewhere in China. Hard to tell by just looking at the front page. Anyway, reinfection could not occur in SARS-CoV-2 infected rhesus macaques. And so they infected rhesus macaques, right, non-human primates, and the monkeys developed symptoms, weight loss, replication, moderate interstitial pneumonia, and then after they recovered, they challenged them, and they were immune to subsequent disease. And the only reason I point this out, of course, it's not humans, but now everyone is worried about these reports coming out of China of reinfection, right? Mm-hmm. And we talked a little bit about this with Ralph, and he talked about the experience with MERS and so forth. But I think this shows this is a virus that is typical in the sense that you get infected, you get immunity, and you're protected. I don't know what's going on in those reports.
If you're a macaque.
If you're
what?
what?
Yes, if you're a macaque.
Yeah, of course, macaques.
The reports, it's questionable whether there's reinfection happening. But by the way, bioarchive now has a yellow banner at the top saying they're receiving a lot of papers about 29 nCoV. They're still calling 2019 nCoV. They're still calling it. Reminder that bioarchive is not peer reviewed, and then they say that these papers shouldn't be regarded as conclusive, guide clinical practice, or be reported in news media as established information. So, if we're news media, then this is not established information.
No, but this is being reported everywhere, right?
Of course it is, yes.
In fact, I spoke to a reporter, BuzzFeed guy this morning. He said, isn't it crazy that there's so much going on social media and most of it's wrong? And I said, well, welcome to the world. But you have to, among that is good stuff that you can find, but it's hard to do. But that's the way it always has been with Twitter and Facebook and other social media. There's a lot of garbage and there's some gems amongst them.
The other quick thing from this paper is that the antibody titers are actually extremely low. Mm-hmm. So in macaque 3, the titers were 1 to 8.
Yeah, it's pretty low.
And that was sufficient to block infection. So it's an interesting, I don't know what that means.
Yeah, Ralph said last time that in MERS, at least, in humans, the antibody titers decline over the next year. And people can be reinfected, but they have mild disease.
Yeah, I think these... how long did they wait before they reinfected them? And
the macaques?
Yeah. I mean, it could be that if you, take a big cohort and you take a bunch of them and reinfect over long periods of time.
Sure.
Well, it's only 4 macaques.
Yeah, it's only 4, yes, because they're expensive.
Yeah.
Right.
Always the problem with non-human primate experiments. I don't know the answer. I don't want to look it up right now, but that's a good question. All right, now we're going to do some email.
Finally, that was the purpose of this show. Let's do email.
Yes, for longtime TWiV listeners, we've traditionally gone through and actually read people's emails on the show because we have 90 plus pages of these. Now we're going to be summarizing, grouping these, and in some cases, not reading any of it, just saying, okay, and these people asked about this, and now we're going to talk about that.
But as usual, the entire text of everyone's email will be over at the show notes, microbe.tv/TWiV. And we're going to do another one on Friday to try and get through. But, I think many of them are very good questions. So we do appreciate them. And there's not many other places where you can, ask a virologist.
Right. And get an answer.
Or a medical student.
And we've also, despite what order they come in, and we're kind of trying to move more urgent or more discussable ones up toward the top. So if you don't hear yours, it's not because it's unimportant. It's just because we didn't quite get to that grouping, and we will.
We'll get to it because none of us are traveling. So
that's right. Nobody's doing anything else. So, all
right. Michael writes, toilet paper equals camouflage for the mummy apocalypse. Obviously. Yes. That's a great reason for it.
And my sister commented that the canned beans were also sold out, so mystery solved.
All right.
That's why everybody's buying up the toilet paper.
It could be. Alan, do you want to take the next?
Sure. So we got a bunch of emails about remdesivir, which is an antiviral. We were just talking about some antivirals that these are being tested worldwide in a few different studies. Scott wrote about remdesivir and chloroquine and sent a link to, I'm not sure if this paper was the one linked above, but another chloroquine paper. Vincent, you've got the clinical trial link for remdesivir. Judy wrote from Perspective in Singapore, and talking about the same thing, remdesivir.
So there are 1, 2, 3, 4, 5 trials.
Ryan also wrote about that.
Trials for remdesivir at clinicaltrials.gov. So this is happening right now. We're going to know, within a reasonable amount of time, what the story is with remdesivir and chloroquine. Let's see, just for the heck of it, let's do a chloroquine search on clinicaltrials.gov. So we'll do. Go ahead.
I did it yesterday. There were 3.
3, okay.
Yeah, and let's see if there's more today.
It could be. This is a rapidly changing situation.
I think there was one in Thailand, one in South Korea, and then one in Minnesota.
There are 5 today.
5 today. Yeah, there you go.
5 today. Well, at that rate, the curve will be.
I think that we will know shortly. And
yes,
if useful, if these are effective, they could be used in more and more people. Especially, Ori, would this be used in a more seriously ill patient? I guess so, right?
So yeah, that's called compassionate use. So that remdesivir is being used widely as a compassionate use drug, but has not been evaluated in terms of any rigorous trial.
So if you do, these are trials ongoing. Even if you do a trial, the FDA would still have to approve it. No, otherwise you could still do compassionate use. Is that right?
Yeah. So I think that essentially for insurance coverage and like for medical legal stuff, the FDA approval is required. Yeah.
Right. So just a quick, I'll just finish up with Stephen, who is a medicinal chemist who commented on the remdesivir, and pharma companies are ramping up, presumably screening all their libraries. And he wondered if there are any data on case fatality rates with HIV or TB co-infection. And I don't think so, but that is a very interesting question that is going to be explored in depth in quite a few countries around the world, I think.
Oh, this is interesting. Yeah, he has some insight into the patent on remdesivir and the different enablements and so forth. So it's worth looking into. But the last point he's talking about, the incentive for making drugs that we've talked about. He says, in my opinion, we need a fire department model for this class of medicines. Most people willingly pay taxes to cover their fire department, even though they hopefully never need it, but when they do, it's there. Antibiotic research and development should be the same. That's a good idea, right?
Yeah, absolutely. We've talked about, that general idea before us. There needs to be some funding mechanism that's not a profit-driven or anything like that. The
other thing that's interesting is that that he points out that remdesivir is parenterally administered, so either through IV or through like an IM injection, whereas faraparavir, which we spoke about earlier, is oral. So you can imagine that being used as an outpatient treatment of the virus.
Yeah, that's a good point. Oh, one more thing here before we leave this group. Ryan said he thought that SARS-CoV-2 is a retrovirus and would have reverse transcriptase, but of course it's not.
No, it's not.
It's a plus-strand RNA virus in the same Baltimore class as poliovirus and flaviviruses. So the genome is plus-stranded and it encodes an RNA-dependent RNA polymerase. So that is a good target for antivirals, RNA-dependent RNA polymerases, proteases, which are encoded in the coronavirus genome. Those are 2 great ones. Enzymes are good targets in infected cells because they typically are there in lower amounts because they're enzymatic. They're catalytic, right?
Right.
All right. So, Rich, can you take Alexandru?
Sure. Alan, you've highlighted a lot in yellow here. Is that your sort of major points?
I think those are the major points, yeah.
Yeah, okay. So I'll adhere to that for the sake of efficiency. So Alexandru writes, asking several questions. I've recently been going down the rabbit hole and kept reading about cytokine storm. I don't know if there is proof for this to happen with this virus, but I could imagine this to be the cause of people fainting. Someone mentioned those in the last podcast. And if so, how could someone stop that? Immunosuppressive modulating drugs, oseltamivir, etc. Let's go through all of his questions and come back to him. Second, what's your opinion on this? They've been working on a vaccine for IBV. What's that?
Infectious
bronchitis. It's not inflammatory bowel disease. It's because I was thinking that at first too. It's infectious bronchitis virus.
Trying to adjust the vaccine for SARS-CoV. Is the time frame they mentioned realistic? I haven't looked at that. An extra link. If this interests you, they're predicting that Seattle is now what Wuhan was on the 1st of December. That's interesting.
So IBV is a poultry pathogen. It's a coronavirus of poultry. So they have a vaccine in development for that. And, they want to just adapt it to this one. Well, it's not going to be quick.
They say 8 to 10 weeks, which
that's ridiculous.
That's crazy.
It's crazy.
Well, that's too... Yeah, this is going to come up a lot because people are sending us a lot of stuff about vaccine candidates and trials, and a lot of people are working on this. And it is true that within weeks, or in some cases within hours, it's possible to produce a vaccine candidate. Mm-hmm. The problem is candidate, and getting from there to actually, does this thing produce antibodies in animal models, multiple animal models of different species, and is it safe in humans, and is it effective in humans? That's what the 18-month time frame is about.
Mm-hmm.
Let's go back to his first question about a cytokine storm.
You remember Ralph talked about that last time, remember? He said NIH has been working on it for years, and they've got the fatality from 50% to 25%. Ori, do you know of any drugs that treat cytokine storms? Is that?
I don't know very much. The only thing that I know that is going on is they have like anti-IL-6, different monoclonal antibodies against cytokines that are used in different clinical contexts.
I see.
Yeah.
For the naive out there, cytokines, correct me if I get this wrong, but cytokines basically are signaling molecules involved in controlling the immune response. And it's a very delicate balance to get that signaling right. And if it gets out of balance in a raging infection, your immune response can be, in a way, overstimulated to the point where it causes severe disease. And so being able to control that to some extent and avoid that cytokine storm is a worthy thing to do.
And that's why you could imagine.
And there are a lot of pathogens, disease conditions where the cytokine storm and the immune response are worse than the original virus or whatever.
And that's why sometimes people use steroids in the context of an infection, which would be counterintuitive, but you want to actually block the inflammatory response at late stages. Yeah,
finding the Goldilocks point in a virus infection or any infection, where you're got enough immunity to deal with the infection, but not so much that it's causing you a problem. That's a challenge. Mm. He says, also, I keep reading that virus binds to furin protein instead of ACE2. What would this mean? That sounds to me like a misunderstanding of what's going on. The furin protein is a protease that is responsible in processing or activating the spike protein for it to do its job. ACE2 is the protein that the spike protein actually binds to. Okay, so I suppose you could say that the furin protein, that is the protease, interacts with the spike protein during the process of infection, but it's not the receptor. The receptor is ACE2.
That's right. That's right. ACE2. There's two of them. There's ACE and ACE2. That's really important, as we'll see for the next one. So, Ori, there's a bunch of questions about ACE blockers. Do you know anything about ACE and ACE2? In terms of blocking, and so ACE2 you don't block, you actually stimulate it, right? Because that's the good ACE,
right? So I actually read a bit about this because it's quite interesting. Because a priori you'd think that an ACE inhibitor would be beneficial in this disease because it could block viral fusion, but actually long-term ACE inhibition leads to upregulation of ACE2 expression, therefore giving more viral opportunities for viral entry.
Can we back up for a minute here and just talk about ACE in general? What are these molecules and what do they do? So
ACE is an enzyme that is, it's called angiotensin converting enzyme. And this is involved in a pathway, a hormonal pathway called the renin-angiotensin-aldosterone pathway. That's important in terms of fluid balance. So inhibitors of angiotensin converting enzyme are used in renal disease, cardiac disease,
blood pressure, right?
Right, as a blocker of blood pressure. There's also effects on remodeling of tissue. So they have like very nice disease modifying effects in heart failure or kidney disease. And many people, including people with diseases that are the highest levels of comorbidity with COVID, such as hypertension, heart failure, and diabetes, are on ACE inhibitors.
But ACE is not the receptor. ACE2 is the receptor. Right? So ACE blockers are not going to help directly block virus infection. But molecules that bind ACE2, and my understanding is you don't actually want to inhibit ACE2 because it has a beneficial role in blood pressure. It reduces, I think it vasodilates. The downstream effect is to vasodilate. So you actually have stimulators of ACE2. And the question is whether they would have an effect on virus because the enzyme active site is separate from where the virus binds. So you would have to have an allosteric effect of those molecules. And we just don't know if that's the case. Does that make sense to everyone?
Yeah, I think. And then the other problem is this like compensatory changes in expression of these proteins or of the expression of the genes over time after being exposed to ACE inhibitors, and that can change susceptibility to disease.
Yeah.
Right. So a lot of this discussion has been stimulated by a letter to the Lancet and subsequent vast amounts of media coverage of said letter, which this is a letter from some folks, Lei Fang, George Karakiolakis, and Michael Roth at a few different institutions, Basel, University Hospital Basel, and University of Thessaloniki. So they point out this phenomenon that ACE inhibitors stimulate, upregulate ACE2, and they hypothesize that this might increase susceptibility and explain why patients with cardiac diseases and hypertension are developing worse illness with SARS-CoV-2. There are no new data in this. This is a letter. This is a hypothesis. It's a reasonable hypothesis, but untested. However, I think the line that's gotten the most attention was they point out that ibuprofen also increases ACE2 expression. And so everybody's been running around saying, oh my God, if you think you have COVID, don't take ibuprofen. There's a good New York Times article talking about the subtleties here. The WHO has unfortunately recommended that has jumped onto this and said, don't take ibuprofen. I think the EU health authorities have come out and said, no, ibuprofen is fine. So there's a tremendous amount of confusion that's been generated about this. I think the important point is these folks were presenting a model, something that ought to be looked into, and it was very reasonable for them to bring that up and something people ought to be thinking about. I think I'm going to guess that they're probably a little bit horrified at how this has been picked up too. I don't know them, but that's a guess.
The American
this is a lot of the discussion.
The American Heart Association released a statement yesterday, which I'll paste in, to not stop taking your ACE inhibitors.
No, oh God, don't stop taking your ACE inhibitors. No.
And very clearly said that there's no evidence for this and that this will essentially is a hypothesis generation paper. Yeah.
Going back to the top of the show, I looked up the quote. The quote is, all models are wrong, but some are useful. And it's attributed to a statistician named George Box.
Good. Yeah, we have mentioned that before.
Yeah.
I don't know if you noticed, Ori, we have a letter here from an MD PhD. He says, but I am just a medical doc now.
Yeah, it's hard to do both.
So, it's funny he says just because PhDs often say, I'm just a PhD. And I always say, don't say just. Right. But now we have a doc saying I'm just a medical doc. Not a, so there he's putting the PhD part, that's pretty funny.
And I'm just a medical student. So
I'm just a medical. We're all just people, right?
Well, yeah, I was going to say, if anything, if this virus teaches us anything, it's going to teach us that we're all just people and we need to get our act together.
All right. We have one from Steve. Oh, Ori, see Steve there?
Yeah, you want me to
see the highlighted part? It's all about hand wash, right, Purell?
Yeah. You want me to read this?
Sure.
I stayed in a psychiatric unit once and observed that the communal hand sanitizer dispensers were emptied by the patients as fast as they could be filled. They really need to find something else to use in units largely occupied by alcoholics.
Yeah.
They inhaled the lot in no time.
Oh, wow.
Yeah, not ideal.
He says basically we should stop shaking hands.
Right. We should go to bowing.
Which is, I think bowing is very cool. I have no problem, but I've stopped shaking hands.
Yeah, I do the foot bump with my grandchildren. Because I like that because it's a dance.
And then he's also got some comments on ACE inhibitors and so forth, which we've talked about. And that ends the ACE2 series.
Yes.
Okay, Goldberg drum. Alan, tell us about a Goldberg drum.
Right, so Jeff writes in about this. He wants to clarify where we're talking about a medRxiv paper showing that the virus stays aloft in a Goldberg drum for 3 hours. And he points out that this seems to be this drum system seems to be a way to keep particles aloft that would normally fall. And he links to a 1958 paper that seems to be the original reference to this, and points out that the 3 hours means it does not mean that SARS-CoV-2 stays aloft for 3 hours. In fact, it's highly unlikely that it would. And references this.
Okay, good. Yeah, because I was listening again to the Barrick, and we didn't know what a Goldberg drum was. So this is good. This is what it is. And this is a paper now that's been published in New England Journal.
Right.
Okay.
And then I'll go ahead and take Chuck's as well.
Yes,
who issues a correction. He says he's a pilot with a major U.S. airline, flies Boeing 787s to South Korea and China until this month. On all Boeing and Airbus aircraft operated by major airlines, cockpit air is supplied directly from either engine bleed air or an electric compressor. The air is fresh, never recirculated, supplied at a pressure slightly higher than that supplied to the passenger cabin. So the cockpit is getting a different air supply than the cabin. So good to know.
Good to know.
In the planes I fly, the air is always totally fresh too, and you've got your choice of the heat being on high or off. So,
but the rest of the plane, it's recirculated, right?
Yeah, in a commercial airliner, the rest of the plane, a lot of the air is recirculated.
As we talked last time, yeah. So Chuck just discovered us, but he said he's going to be a regular listener.
Yeah.
Guess if you're not
especially since he's got time on his hands.
But I tell you, these 787s are awesome. I flew one from San Francisco to Singapore. Man, they are really, and the air is really good. It's much better than other planes, but because I think they do less recirculation, maybe, or I've heard that. Maybe. But it's a wonderfully comfortable plane, though. Really nice. I really appreciate it. So hopefully everyone will get back flying sometime this year.
Yeah.
All right, now we have a mask series, right?
Yeah.
Yes.
Let's see. Well, what's the bottom line here? Oh, there's a nice...
so...
Go ahead.
We ended up dancing around the mask point because we cited two papers, one of which said masks don't do squat for the general public, and the other said that masks might be helpful for the general public, and both of those were published in the same CDC journal at around the same time. So I think, and people are pointing out, well, maybe they are helpful, and if they're not helpful, then why do medical professionals wear them? To which I would answer that medical professionals are up in people's faces who are sick. So it's a different level of, if you're being actively coughed on, then you certainly want a mask on, and that's part of your general equipment. For people walking around in public, I think it remains an open question. for most people these days, it's going to be a moot point because you can't buy them. They're right next to the toilet paper. And I think, I don't know, maybe if we get to a point where everybody else is wearing them, I'll put one on too if I can get it.
And first of all, people say, please clarify if you're talking about surgical masks and N95s, right? So,
and in fact, we got a couple of emails about additional ones I wasn't even aware of, Cured antiviral face masks, which apparently have a layer of ascorbic acid or something that inactivate viruses. And then somebody else asked about respirators like painters use, like the half face respirator. I don't know.
Or you have any thoughts on the face mask? Surgical masks is what they give healthcare workers, right?
Yeah. If you're sick and sneezing, wear a mask. If not, save them for the healthcare workers. So essentially, the PPE supply is in an extreme shortage in New York.
PPE being personal protective equipment.
Correct.
Right?
So masks, goggles, and gowns. And generally, they're being wasted in the general public who's not sick, whereas healthcare workers who have a very high chance of getting infected with a droplet from a patient need them. And that in my mind is the strongest argument that there is against not wearing a mask.
Yeah.
So if we had enough masks for everyone, then people could wear them, right? So what I understand is that the supply is an issue, but also if you don't wear them properly, they're not going to help you. They do get contaminated. You have to know how to take them off and so forth without contaminating yourself. But as Steve Morse said yesterday, in many Asian countries, many people wear them. They have for years. It's part of the culture. Yeah. And so, there's this general thought that maybe it does help. And from what the literature says, it can help under the right conditions if they're used properly. But we simply don't have any. Here, we got a request from, I think, the head of medicine.
Mm-hmm.
If you have any extra, give them to us, right?
Yeah. No, and the other thing, in the hospital teleconference this morning, they said that they're walking around the hospital and they actually see healthcare workers in the hospital with them only around their mouth and not around the nose. So that's not useful. So they admonished everybody about that. So if you wear them right.
Here's the mask question no one is asking from Dominic. Why do you continue to ignore the efficacy of Middle Ages plague doctor masks?
Yes.
Surely the elongated beak places additional distance between us and anyone who is infected. And the flowers and herbs packed into the beak will act as an effective filter. Tongue firmly in cheek.
Have you guys seen the videos of people walking around with large wooden discs around them that are 6 feet long?
No.
So people can't get close to you.
It's to keep people 2 meters away.
Yeah.
How long can masks be reused? Does anyone know? You're supposed to dispose them, right, Ori?
Yeah.
Have you worn one, by the way, in your brief tenure in your rotations? Did you ever wear a mask?
Yeah.
And what purpose was that? A patient, or don't you put them on the patient usually?
So the patients wear them, and then we would wear them depending on the precautions. So, for example, for influenza patients or for patients with communicable diseases, there are usually signs on the door that says the level of precaution, and then you take a mask from outside or a gown from outside, and you put them on before you go in.
I see.
But a lot of times, at that point, essentially, anytime you sent out a test to test for the flu or this this biofire panel, people would automatically be placed on precautions, but I don't think that's the case now. Mm-hmm. So we were wearing a lot of masks. Yeah.
So someone asked,
and you would wear it once and dispose of it.
Correct.
That's the point. So someone said, "Can you reuse a mask?" But you don't want to do that, right?
Well, the chances of, on the outside of the mask, there are droplets, right?
Yeah.
So you have to be pretty careful.
Yeah. Although if we're talking about the general public and we get to a point where the mask supply is adequate and people are still thinking about wearing them, then it's, probably do what you want. But yes, if you're in the high exposure environment where you actually have a reason to be wearing the mask these days, then that is a disposable item that needs to be thrown away.
Mm-hmm.
Yeah, I think given unlimited quantity, you could wear them. And subways at this point would be a good idea. Airplanes, if we were flying, right? All those situations. If you're not around people, I don't see why you'd wear it. I saw a woman driving on the highway today alone in her car, and she was wearing a surgical mask. So I wonder maybe you should just save it till you get out and go among people, right?
You don't have to worry about your breath smelling bad, though. There's a lot of benefits. Yeah.
So we don't know if these antiviral face masks actually work, right? They're impregnated with citric acid.
Right.
If that's true, the inner layers are coated with copper and zinc ions. Now, if that's true, the copper would be very antiviral. I don't know actually do they exist? Does anyone know that?
He says Curad antiviral face masks, and or she,
Jackie, yeah.
Not sure. So yeah, I guess that's a product that exists, or at least existed on store shelves at some point.
Yeah, and there's nothing on Amazon. They're currently unavailable. Yeah.
Well, no, right.
Yeah. Michael Schmidt said some companies are developing copper impregnated surgical masks because copper is very good at inactivating coronavirus and other viruses. But he said they're under development and they have to be tested to see how much copper you need to put in and so forth. Right. And of course, if they get very expensive, because copper is expensive, depending on how much copper is, then it's a problem. Right. Are there such things as reusable face masks? I guess that more substantial ones with pumps and respirators are reusable, right? Is it
correct? Yeah.
Well, and the painter's masks have replaceable filters on them.
Yeah.
Mm-hmm. What's an elastomeric respirator? Does anyone
That's
know?
the one. That's the painter's mask.
3M model 7502 half face with pink disc filters. It seems that would stop droplets, right?
Yeah, those are designed for volatile organics. So they're going to stop pretty much everything except air, I think.
Do they have carbon in them for the organics?
I think they usually have a carbon filter for the organics, and they may have interchangeable filters to filter out just particles if you don't need the C filter. I'm not sure.
So it says that it provides the same protection as a disposable N95, but healthcare workers use it more because it's perceived to have a better fit if it's available.
Right.
All right. So the gist of many of these emails on masks are that many people do not believe that they're not protective, and I think there are data, as Alan said, that they can be if used properly. But right now you can't get them, and you should not buy them because they should be going to healthcare people. But in the next pandemic, I think we should have a stockpile of face masks. Of
course, yeah.
And they should be part of what is released, and they should teach people how to wear them, to put them on and take them off, right? In fact, Gerald Block, who's an MD in Hawaii, also mentions this, but he also says, why don't you explain why this can't be a manufactured virus? Well, we've done that on a previous show.
Right.
And so I think we did a nice detailed explanation of that. I don't remember the number, but it was done, Gerald. So check that out.
And the bottom line is that the sequence of the virus that's out there is simply too different in particular ways to be something that any human would create.
Yeah.
It's just not at all within the realm of reason. He also says up at the top, I don't buy the assertion that N95 masks do not protect the person wearing them from COVID-19. Simply put, if that were the case, us doctors would not be wearing them. Right. I don't think nobody, or at least we aren't asserting that N95 masks don't protect a person wearing them, in particular in a clinical situation where you're down close to a person who is exhaling respiratory droplets on you. Certainly, in that case, an N95 mask properly worn is going to provide some protection. He says, I don't think all the PPE is to protect the patients from our germs. No, it is to protect you. Under some circumstances, however, which we already pointed out, if you're sick, a surgical mask will help prevent you spreading it to other individuals. So
now we have some questions on serology. Per. Why is the case number dropping in China and South Korea? Is the virus being contained? Is population immunity on the rise? Serology could provide the answer. Yes, I'm sure we're going to start doing serological studies. As Ralph pointed out last time, though, China does not plan to do any.
Right.
And he suggested it was because they don't want us to know exactly how many people have been infected. I don't know. What do you think about that? Is that a reasonable conclusion? I suspect we'll see some serology at some point.
I think we'll see a lot of serology from some countries. I would hope that we'd see a lot of serology from the U.S. and a lot of other countries. Basically, what China wants to do, I don't really care. It's their business. As long as we get, get good data from somewhere with a fairly large population where we can chronicle both how much disease, how severe it was, and do all the epidemiology combined with serology so that we have a good number for how many people were actually infected.
He closes, there must be a reason that countries are reluctant to start or report serosurveys. Is this because it would reveal that the whole exercise on containment has failed? I cannot think of a scientific reason for not doing serology, only political.
I'll just add two logistical concerns, which are actually getting a working serology test, right? So first of all, it takes 5 to 7 days to get antibodies that are to become antibody positive. And then there's a lot of cross-reactivity with existing coronaviruses. So we could have a serology test out there that is detecting antibodies against some common cold coronaviruses, and that won't be very useful.
Yeah. Right. Yeah, you have to get the antigen, the important antigen, and then you have to immunize animals and get a very specific antibody, right? Mm-hmm. And you have to, qualify all of that before you do the study, right?
Yeah.
Right now, it seems to me that there are bigger fish to fry. Okay.
Yes.
And this is something that requires a significant effort.
That said, an antibody test would be useful for combating the epidemic as well. This is not just idle speculation. This is, right now we're doing a PCR test, and that's going to tell you if you have viral RNA circulating, which means you've got probably an active infection. But it would be very useful, I think, to know if somebody has been infected. And I'm sure there are people working on antibody tests. And, as Ori said, these are not trivial to develop, but there is an established procedure for developing them.
Yeah.
And this is something that should become widely available very, very soon, I would think.
And the most important reason for it to be useful now is that it will be able to test whether healthcare workers are immune. Mm-hmm. So the idea being that right now, if you're exposed and you have symptoms, then you're pulled out of the rotation in the hospital. But if we could figure out which people could be exposed without risk of reinfection or infection at all, then you wouldn't have to pull them, and then you would have less workforce problems.
Yes.
So Jake suggests that you could, like you said, you could identify people who have been infected, and then you would know that they are no longer at risk, right? And they could go back to work and so forth.
That's exactly the point.
And they could avoid contact with people who are at high risk.
Could we think about issuing COVID-19 recovered certificates?
Right.
I think this is a logistical nightmare.
No, I wouldn't go with certificates, but I would love to see some serology.
Well, and also you could have antibodies but also be shedding virus. Do you think that there could be asymptomatic shedding in serology positive patients?
Mm-hmm.
Sure.
Yeah, there could be. I don't know how you would implement this. Serological surveys are usually done to, estimate the fraction of a population that was infected, right, after an outbreak. But are these done on healthcare workers to see if they're immune to something, Ori? Is that routinely done for other infections? Or
well, you could imagine. So
or do you just vaccinate everybody, right?
No, so generally they actually will check titers even for existing vaccines to make sure that you actually have immunity.
Oh, okay.
But in this case, it's extremely important to make sure that they're pulling people out of the rotation as little as possible, essentially, to go back in.
It occurs to me that there may actually be people out there for whom this term, even serology, is a mystery. Mm-hmm. This involves taking blood, right, and testing it to see whether you have antibodies to the virus. If you have antibodies to the virus, that means you have been exposed and mounted an immune response. And it can be even more sophisticated than that and tell you something like how recently you were exposed, but that's the basic story.
Yeah.
And it occurs to me that if you're going to do a serosurvey, I mean, from a practical point of view, from a research point of view, how do you do this? Do you, in an organized fashion, go around and ask a whole bunch of people to give blood, or do you go to blood banks of one sort or another? Where do you get your samples? How are they?
You could
do it
both.
either way.
You could do both. A blood bank is a great way. You know, our friend Eric Delwart could do that sort of thing because they have lots of blood. But you can also design a survey, say in a big city, you want to know what fraction of the population. You design it with clearly selected individuals, not just, you know, we're going to take 100 people from Columbia Hospital. You want to sample different areas and make sure you get different age groups and all sorts of things so that you can then extrapolate to the general population using statistics, right?
Mm-hmm. Right.
Well, you don't have to check everyone for sure.
I've been thinking over and over again, the old saw that there are 3 types of lies. Lies, damn lies, and statistics.
Damn lies and statistics.
And, you know, it's interesting. A number of these people are saying the same thing. Another, Amy has said to do serology so that we can identify a workforce that's safe from adding to community spread. That's exactly what we've been talking about. So these are just coming online, I understand, and so that'll be used more and more, I think.
They don't have them yet.
Here they don't have
them yet. Yeah.
Yeah, but I think I've read that China is already doing some serology in other countries as well, even though they won't tell us.
Right.
I would really like to have some kind of accurate number on what fraction of the population has actually been infected.
Yes.
You know, that to me is one of the big missing numbers.
And that would also have a huge impact on all this quarantining and shutting down the economy.
Absolutely.
Well, because if it turns out that a huge number of people are already exposed and immune, then you can start lifting some of this. And if it turns out that they're not, then it means that you can continue the quarantine and maybe not get out of control.
Yeah. Steve Moore said yesterday he thinks 70% seropositive is needed for herd immunity. And
that's what Ralph said, too.
I think you're going to get that very quickly.
Yeah.
If it's not already at that point.
I mean, certainly in China, it may be close to that. In
some
areas,
countries, it may be. Yeah.
So we have an interesting email from Raphael, who's a psychiatrist, who says, should we be social distancing in Portland since we know what's going on in Seattle? And I think we should be doing this everywhere. There are cases throughout the U.S. Even if there's no one in your region, it's there. You just don't know about it. So
the biggest problem right now in the U.S. is that we don't know.
Mm-hmm.
Yeah.
I mean, so the safest assumption is that every place should be shutting down public stuff until we have enough testing to be able to figure out where all it is.
Yep.
And I noticed that Raphael is a psychiatrist, and it gives me an opportunity to say, you know, this is a great opportunity to figure out how to keep your head together. Okay.
Yes.
I've discovered, for example, that there is a gardener in me somewhere. And if I go out and don't listen to music, don't listen to podcasts, just listen to the birdies and dig the weeds out of my garden and make it look nice, it has a wonderfully therapeutic effect. There are ways that you can use your time that are beneficial in this crisis.
Birds are remarkably relaxing as long as they don't wake you up at 5:30 a.m. when the light comes out.
And of course, then I find myself thinking that during what was it that killed all the crows? West Nile. When West Nile came across, it killed 90% of the crows. So the birdies don't help me at all.
So Sarah wants to know, and this may be one for you, Ori, you know, that she's thinking of getting pregnant. Is that a bad idea?
I don't know. So I will just say that. I have 2 friends who are pregnant now, who are about 6 months and starting 3rd trimester. They're both nervous, and their doctors are saying that there is no data. So there's nothing to say. Don't get infected.
Yeah. But of course, we know with influenza, it's not good to get infected during a pregnancy, right?
Yeah. And there was one paper that I saw that I'll put the link in the show notes that infants who are born to positive mothers were not positive.
You mean SARS-CoV-2?
Correct. Yes, but it's unclear whether that was. It's unclear at what point they tested, whether it was immediately after birth and they just separated them. Obviously, if they put them back with the mother, there will be transmission that way.
I'm just concerned that if you have a high cytokine level, that could negatively impact the developing fetus that in ways we don't know, right? Because these cytokines are soluble; they go everywhere.
Yeah, I mean, but it's just it's an underlying medical condition that could make the infection worse for mom or for baby, and we just don't have the data at this point to know. The interesting thing is the hospital is not excusing healthcare workers who are pregnant from caring from COVID patients. So they're making a choice that it is not so scary.
Interesting.
Yeah.
I don't know. If it were up to me, I would wait because of my cytokine concerns, but I don't know how founded they are because, as Ori said, we don't have any data. But she says it's interesting. What about across all people getting pregnant at a population level?
I hope all people don't get pregnant at a population level.
Well, it's been proposed.
Fortunately, I don't think that's how it works.
There's going to be a boom out of this.
Yeah, that's right.
The coronials.
Yeah.
Coronials.
I think the coronials. I like that.
Oh, my gosh. Did you just make that up or did you hear?
No, somebody laid that on me.
That's good. Yeah, I think if you're not currently pregnant, it might be good to wait at least a couple of months and just see how this all shakes out.
Yeah, I would wait.
If you're already pregnant, try and limit your exposure. If you're a pregnant healthcare worker, try and limit your exposure. But do what you can.
And it's a similar vein. The next email from Glynis wants to know if she's got a wedding planned for October. And her dad has got diabetes and COPD. Mother has got heart issues. There are other people who are ill of all sorts, right? Should I go ahead and do this? October.
Who knows?
Yeah.
A honeymoon is planned in Rome.
Who knows?
Maybe.
I think this is a big question. How long is this going to go on? We don't know.
Yeah.
I don't think it's going to be through October, frankly. I have a guess that it's going to be July extinction based only on SARS-1. I could be wrong, but it's just tough because you have to plan ahead of time. And I realize, I think if you want to be safe, you should just wait and do it for next year. Maybe you could plan it for the spring and it should be over by then.
On the other hand, flights are really cheap now. So
I would say on the honeymoon, wait to book that. Because the sales after this is over are going to be, I suspect, pretty deep. Please come fly. Please come stay in Rome. And you will probably even do better financially by waiting. There's not much downside to waiting on booking travel at this point. In terms of the wedding and stuff you've already put deposits on, that's a really tough call. I don't know. ASV was just canceled, which is happening in June.
Yeah.
Big meeting.
And the BAT meeting afterwards?
Canceled. So I've traveled to Germany in October. I'm not sure that's going to happen. It's a meeting, and I don't know. It's still up in the air, but if you could postpone it, that's what I do. And then she asks, I've offered to go out shopping for people who are more at risk. What advice would you have? So I told someone today, a reporter, that I went to the supermarket Sunday morning at like 7 a.m. and there was nobody there. Mm-hmm. So if you pick hours where there are not many people around, that's a good start. Stay away from people who are obviously sick, and, everything you pick up, I would treat as potentially contaminated, right? A jar of sauce or a milk carton. And I know people who bring them home and wipe them down. You could do that, or you could put them away and the virus infectivity will decay for a while if you don't need to use them for a few days. But I would just keep, the credit card, the little button thing at the checkout where you put your code in, all of that is potentially contaminated. So just make sure you wash your hands extensively, right?
In one of the masks that we cited last episode, another thing that correlated pretty strongly with people being infected from SARS-1 was washing their hands when they came home.
Yeah.
Yeah.
Yeah. My wife went to Costco the other day, and you've probably experienced this, but there was a line out, not because the store was crowded, but because they were limiting the population density in the store.
Right.
Yeah.
And not letting too many people in at once. And the line, they insisted that people be 6 feet apart.
No, were they maintaining that?
Yeah.
Because that's what Valeria Cagno told me a couple of weeks ago. In Italy, they were limiting people in stores, but they were just queuing up right next to each other outside, which, of course, is bad. Don't do that. Can we keep going, Alan? You okay for another?
Yeah, I think we can keep going. The only reason I put that in there was I thought it would take us 2 hours to get to this point. So let's keep trudging along.
All right. Someone can read some of these yellow ones. Rich, can you read those yellow parts?
Sure. This is from Leah. I'm a school nurse by the way, thanks for doing all this, Alan. This is great.
Mm-hmm. Oh, sure.
Leah writes, I'm a school nurse practitioner at a small school in Philadelphia. Up until this past year, I've been an emergency room nurse in Philadelphia. During my 10-plus years working in the ER, I have witnessed overcrowding and the struggles associated with providing care to sick patients in a system that isn't equipped for the volume of patients or the severity of illnesses seen typically without a pandemic. With the recent closure of another large hospital in Philadelphia, I don't believe we're equipped or prepared for this. During the past 2 weeks, I've seen an influx of children sent to my office for mild upper respiratory symptoms. Children between the ages of 5 to 12 have been complaining of a number of symptoms, including cough, runny or stuffy nose, sore throat, headache, body ache, respectively. I have not had any students with fever or severe symptoms that would warrant being sent home or prompt me to order additional testing. However, everything I'm reading and hearing indicates that children are likely to have very mild symptoms and may even appear asymptomatic. While I do take a moment to provide reassurance and hand hygiene and infection control education to each student, I am not worried so much about them when it comes to the coronavirus. I am very worried, however, for the grandparents with chronic obstructive pulmonary disease, diabetes, hypertension, and other comorbidity that are picking up their grandkids from school at the end of the day. While these children that have COVID-19 are not presenting any differently from common colds, they are able to contribute to the transmission of the virus to vulnerable populations, especially as they are not always polite when it comes to their respiratory secretions and are always touching things. I'm relieved that the schools are closing for the next couple of weeks in order to promote social distancing, but also now concerned because many parents still will have to work, may rely on older adult relatives to provide childcare during this time. Absolutely all good points. And I think my wife and I are confronting this very issue right now. At what point do we limit contact even with our grandchildren? This has been, so far for us, the pandemic has been delightful because everybody's on spring break. My daughter and her family lives a 5-minute walk from here. We've been having dinner together every night. Okay, and watching Star Trek, and I figure, with we don't have a whole lot of contact with the rest of the world, and we're all healthy and all that kind of stuff, so we're probably all cool. But we were just having this conversation this morning. The children are going to be wonderful vectors for transmitting the disease to at-risk individuals, in particular their grandparents, because there's a lot of smooching that goes on.
This is why they closed schools. Right?
Absolutely.
I mean, up until Sunday, the mayor of New York didn't want to close the schools. 1 million plus kids doing exactly this, bringing virus home to older people. So, and I don't think it's for the next couple of weeks. I think it's for the rest of the school year, frankly.
Oh, absolutely.
Yeah.
But the question is now, even if the kids were infected on the last day of school, you have up to a 2-week period where they could still be incubating and not know it. So you got to be really careful. I don't know what to say.
They're gonna, I don't know. I would be considering things like, like we've already discussed, of limiting how many people go to the store. It's no longer a family outing, I don't think. Okay. And making sure that the kids practice the best hygiene they can. I mean, we're doing this now where when the kids come over, first thing anybody steps in the door, you wash your hands. Okay. We don't do a whole lot of smooching anymore. We have a couple of yucks and a foot bump, So this is an important level of awareness, in particular, grandkids and grandparents. I think that's an important point.
Alan, you want to take
Leah's story? The kids coming in over the past 2 weeks, I think, is scarier than the graph in the modeling study that we led off with.
Yeah,
and it could be that the serology ultimately is going to teach us that a lot of these kids were infected.
Yeah.
And by the way, I want to emphasize her point about, and everybody has this on mind, without school, some parents still have to work. We got new issues with what are we going to do about childcare during that time. These are all important challenges.
Yes.
Can you take Leah's, Alan? I'm sorry. Kate. Sorry, Kate.
Wonders basically if closing everything is a good idea. Washington State, is the reaction warranted or is it overkill? And I would say that I think it's warranted.
Yeah, I think it is, and I think in many parts of the country, people don't think it is, and for whatever reason. Yes, we need to do this now.
The only possible downside is economic. I realize that that's very considerable, but making the wrong judgment in this case is going to be many times more disastrous, both in terms of people's lives and the economy. Okay.
Well,
and all of the experience so far says you need to do this.
I mean, one of the big things with closing the schools is, especially in a big city like New York, but really everywhere, there are a lot of kids who school is a refuge. And it's a source of, maybe their only meal for the day. And, it's a huge part of the social support system. So measures like that are drastic. And telling everybody to stay at home, this has psychiatric consequences for a lot of folks. This is an environment that's going to lead to a lot of drinking, to a lot of depression, probably to a lot of suicide. These are horrible outcomes, and these are downsides of shutting all these public services. But I think, given the unknown but large magnitude of what we're dealing with at this point, this is the only sane thing to do right now, is to say, look, right now, since we don't know how big this thing is and we don't know where it is, we got to shut everything down until we figure it out.
And I think also, so far, the experience from pockets of this disease in other countries supports the notion that without these sorts of measures, it can get really out of hand and cause a lot of problems. Okay, so there is evidence to say that the kinds of measures we're talking about are beneficial in this circumstance.
And people don't do it on their own without government mandates. I mean, the bars were full Saturday night when I walked by.
Really?
Near my apartment. So you just it has to be closed down. Yeah.
We have an interesting email from Alec, who has some pandemic experience, having worked in hospitals through SARS, H1N1 MERS, all the other flu season pandemics in Asia and the Middle East. I've seen how much of a strain they put on a healthcare system, and in particular ICUs. In most hospitals, they comprise less than 10% of the bed total, and having to increase or double up on ICU is not an easy task. Equipment requirement, room configurations of ICU rooms are different from normal hospital rooms. I think we talked about that the other day with Ori.
Mm-hmm.
We were short of ventilators during the MERS crisis. We had to take old ones out of storage and refurbish them. These are some of the challenges countries like Italy are facing. Yeah, that's all true. All right. Rich, what does that Bob have to say here?
So this is about the earliest known case. He cites a Guardian article.
Oh, that's the one we had up ahead before. We talked about the same article, right?
Right.
Same article. What is Luke talking about, Rich?
wondering, this is the old question of could the virus mutate and become deadlier? Is the possibility of a secondary mutation a reality? Could COVID-19 mutate and become as infectious as measles, or start becoming more deadly to its more healthier hosts? If a secondary mutation is possible, is there a mutation that is common in coronaviruses that may become evident? Basically, I'm going to say no. This is there's no evidence that this is going to be an issue. This comes up all the time about mutations making a virus more virulent, and I can't think of a situation where this has proven to be true, and there's no reason to suspect that it would happen in this case. Fair enough?
Yep.
Agreed.
Fair enough. Yep.
The mutation rate of coronaviruses is lower than other RNA viruses. Is that correct? Yeah.
Yes, it is, because it has a repair enzyme.
Yeah.
I mean, this virus is, in our experience, we've never seen a human virus become more virulent in the course of its passage through humanity. If anything, it becomes less over long periods of time. It could change its transmissibility, but I think that happened early on in the evolution of this particular virus, and now it's quite transmissible, and so there's no selection pressure for it to become any more. It's all about selection pressure, right? And if you think about it, what would be the selection to kill people quicker? Doesn't make a lot of sense, but to transmit could be if it's not very good at it, but this one is already
This is a virus that right now, this virus is doing great.
It is really efficient.
I mean, it's everywhere. It's infected the world, so it's got no pressure to change anything.
Yeah, the models out there, and the best one is the myxoma in Australia, is that over time, the virus and the host tend to sort of moderate to reach a moderate equilibrium. And I would also say that one thing, we've touched on this before, but one thing I think that is confusing for the population is that they're used to discussions of influenza changing and you need to update the vaccines. That's different. It's not becoming more virulent. It's changes in its coat that makes it evade the existing immunity, and so it's not becoming more virulent. And there's no evidence that these coronaviruses do that same thing. That's a pretty unique flu-y thing.
Alan, can you summarize Paul's for us?
Sure. Paul is a biosafety officer at University of Hong Kong. Says he's been working as biosafety officer for more than 10 years. It says, I knew my university would want to think about control measures, and some researchers would begin to work on SARS-CoV-2. Whenever work begins on a new pathogen, you have to go to first principles in biosafety to decide broadly what level of precautions people will need to take. So by way of comparison, I looked around for papers about the other already known coronaviruses that have been circulating for years and came across a preprint, which he provides a link to. Anyway, I would currently advise any deliberate culture with SARS-CoV-2 needs to take place at biosafety level 3, as it's a pathogen with moderate virulence for many, but very high potential for community impact if it escapes, where no prophylaxis or specific treatment is available. In 18 months' time or so, when we live in a world where there are now 5 common circulating human coronaviruses, enough people have seroconverted to make transmission unlikely, and we finally have the first effective coronavirus vaccine, there will come a point, I suspect, when SARS-CoV-2 will be downgraded to biosafety level 2, joining the 4 other circulating coronaviruses that nobody ever talks about.
I think that's right. Here, you have to do it in BSL-3. It's quite clear SARS was BSL-3 as well. In fact, interestingly, after the SARS-1 outbreak, after it had stopped everywhere, there was a small outbreak in a lab in China, which was because they were working with it under BSL-2 conditions. And so that told us we had to change.
So do you think that these coronaviruses initially, when they jumped to the human population, had such a severe outbreak, and that we just, for this coronavirus, we don't have an immune response, and therefore...
Yeah, sorry.
No.
That was Ralph's hypothesis,
right?
Which I thought was really interesting.
Yeah, he
says those came in somewhere between 100 and 800 years ago, so we don't know, but it's quite possible that they were and still are just as pathogenic and transmissible as this thing. but that once you reach the appropriate level of immunity in the population, and the newly infected people are the children who don't experience as drastic a disease, then you reach a new equilibrium that's tolerable.
Very cool.
Yeah. Attention, very writers of fiction.
Yeah,
we're learning a lot from this.
So Eliud is offering to send our police friend Cindy in New England some gloves or masks.
I loved that letter.
Many people loved it. Her PPE is gloves and a Glock too.
Yes.
That's just great. Thank you so much. It's amazing to hear your podcast and be able to laugh and understand the situation rather than hear the media that thinks they know virus. Well, we've always been here. We've been here for 10 years, and we're going to be here as long as possible. So if you never need virus information, you know where to turn. Ori, you want to summarize Louise's for us?
Sure. He says, I have one question about the fecal-oral transmission route of coronavirus in bats or in humans. I would like to know if enveloped viruses, such as coronavirus, can be resistant to gastric juice. What is it in the root of coronavirus, or what is the root of coronavirus in a fecal-oral infection? So Vincent, you might be able to.
Yeah. So these
enteroviruses.
So there are many known enteric coronaviruses of various animals, particularly pigs. They're enteric infections, even though they're enveloped. It was always the interesting envelope virus because, yes, traditionally we say envelope viruses are not going to make it through the stomach, and certainly influenza virus doesn't, and many others. But there's something about the way it's constructed that allows it to pass through the stomach. You swallow it, it passes through the stomach and get into the intestine. For SARS-1, there was even intestinal replication and fecal shedding in an infectious way. And the contribution to transmission, we don't know for this particular one, but it's clearly there. And why these can survive, I don't know. It's an interesting problem.
With enough mucus, anything is possible.
With enough mucus. So the next one, Alan, what do we got here from David?
So David sends a link to a paper on possibly being 2 strains of SARS-CoV-2. This has I've seen this pop up.
So these are just basically single nucleotide polymorphisms, right? That are
This is the paper where they were talking about an S type and an L type, and one maybe being more pathogenic than another, and blah 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 blah. Basically, yes, there is variation in the virus. It doesn't necessarily mean anything.
No, they have no evidence that it's of functionally any importance at all. No. But he does say, this virus has now conducted many human-to-human transmissions. Have we discovered mutations that enhance transmission? Welcome to evolution. Well, the problem is you need to have the virus before it started transmitting efficiently among people, right? And we don't have that, and I'm not sure we ever will. But then maybe you could look at it and see what changes occur. And I'm not sure you could ever do anything about it because you can't infect people, and we don't have a good animal model for transmission. So, unfortunately, we won't be able to know about that. Rich, can you do Frank's?
I'm just going to do the highlighted bit. How likely is it that I will catch COVID-19 just by walking on the sidewalks of Manhattan? And I would say that depends on how many people you're walking with. I think that if there's a reasonably low density based on what I know, there's a reasonably low density of people walking around, and you're not bumping into people and they're not coughing on you and that kind of stuff, should be okay. I think you can go take a walk.
Yeah,
I think you can go down to Central Park and take a walk.
If someone walks by and coughs in your face, you could get it, but you should avoid people. Or do you walk around New York at all?
I have been walking around, staying 6 feet away from people and trying not to touch anything.
Yeah.
But even in the shelter in place, that order that might come through, you would still be able to walk around. You just couldn't congregate with people.
Yeah.
Like you couldn't be with another person.
I mean, you have to be able to go buy groceries at some point,
correct?
Right?
Yeah.
And all that stuff.
The data say that in terms of catching it from somebody, from what we understand, somebody breathing out or coughing out respiratory droplets is only going to be, they're going to drop to the ground within about 6 feet of you. So if you stay out of that radius, you are reasonably safe, it would seem.
All right, we have one from Fernando, who asks, is there any chance a person testing positive for flu masks the possibility that it also contracted SARS-CoV-2? So I think that if someone has respiratory symptoms, you don't know if it's influenza or SARS-CoV-2, especially now that we're in the middle of a pandemic. You should test for both, right? You should have a flu test and a SARS-CoV-2. I don't know in what order you would do it. What do you think, Ori? Would they do both at the same time?
I think that the way to think about this is that we have a limited number of tests. So usually so the reason that we test for flu is because we have a drug that can help reduce the severity of the flu.
I see.
If you're flu negative and you have severe respiratory symptoms, in that case, it might be worthwhile to test for COVID or SARS-CoV, but then at that point, remember that there's no change in management because we don't have a drug, so it would be more to limit exposure and contact trace. But we're past that point, at least in New York City, they're not contact tracing anymore. So the expectation is that there's widespread transmission in the community.
Right. So the question is, if the patient tests positive for flu, do you then not test for SARS-CoV-2?
Generally not, as we're still limited.
Yeah. So they could be infected with both, but if you've gotten to flu, then you are treated as if it's flu.
Right, because then you could potentially reduce symptoms by giving Tamiflu. I mean, in an ideal world, you'd have the SARS-CoV-2 test with the flu test, and you would test for everything at once. But if there's a limited number of SARS-CoV tests, you can't do that.
Right.
So you're saying...
Am I correct that there's a flu test that's basically a dipstick test that you can do on site when not very invasive?
Yes.
So that's a breeze, relatively. Or a SARS-CoV-2, you got to take a sample, you got to send it out, you got to get it tested. So, and the testing is limited. Flu's more straightforward. Is that correct?
Yep.
Yeah, Steve Morse talked about that yesterday. He said if you're infected, it's pretty good at detecting that, but if you're not, it's often you can get false positives.
We had that wonderful letter last time from the woman.
Yes.
Who said her bottom line was, if you test positive for the flu, you have the flu.
You do. And Fernando wants recommendations for grandparents taking care of the grandchildren. I think Rich we talked about that up top, right, earlier?
Yep.
Alan, can you do the Iceland report?
Sure. So I've got to scroll down for the pronunciation. Joi writes from Iceland. Iceland may not be in the news often, except for the occasional volcanic eruption or bank collapse. Something very newsworthy just happened here regarding coronavirus, with a large group of people, including some healthcare workers, having been infected with SARS-CoV-2 on a skiing trip in the Alps, and subsequently bringing the disease to Iceland. Iceland now holds the world record for most COVID cases per capita. Carrie Stephenson, a geneticist who frequently appears in media, Genetics, just announced the company would be launching a free public screening for anyone showing symptoms of COVID-19. But Iceland's Data Protection Authority said in a statement this screening would not be connected with any data that could be traced to a specific person. We already have genome sequences of all inhabitants. Iceland is a very strange place. This means that, in a good way, this means that there is going to be a free and open screening here in Iceland, but we will not pair the test results with genome sequences in order to learn more. I am now doubting my morals, since I very much feel like this is an opportunity wasted to identify some genetic variants that could be important in the infection of SARS coronavirus too. What do you guys think?
Totally. If you could pair the infection with the genome data, that would be awesome.
I think it would be, if there was a way to do this reliably, to make the tests traceable, but not right now. If you could encrypt that link and store it away, not to be opened until next year or something. It would be great for somebody to be able to do a retrospective study. On the other hand, we would expect to get serology tests available by that time, and you could just go back and do this retrospectively that way.
Yeah, I do think you should pair the data, though. It would be really a waste not to. It would be too bad.
Mm-hmm.
But I understand the reasons for not doing it too. So yeah, tough call.
That looks like about as far as you got with your highlighting. Is that right, Alan?
Yeah. I started at the bottom, and so there's going to be now a big gap, and then I pick up again down closer to the bottom. Sorry for that.
No worries. All right. Well, let me just do
On the other hand, we're almost 2 hours.
Yeah, we will stop in a couple of minutes. Let me just do a couple more. So Ben is from San Diego, where he wants to know if you can sterilize a N95 with UVC. He wants to build a hacked DIY sterilizer. 265 nanometers, 10 minutes. Who knows? I think these things are so porous that UV is not going to get into every part of it, and I'm not sure that would be a good idea.
I was just imagining this when we were talking about sterilizing your groceries when you brought them home, and I was thinking about a future home that has something that looks like a microwave in it, where you can nuke everything you bring home. But even that's not going to be real effective because there's always going to be some stuff in shadow, you know?
I think so, yeah.
I mean, it'll maybe reduce it. After all, they have these robots that march around emergency rooms spraying UV everywhere.
Yep.
Okay. So, it's probably better than nothing.
On the other hand, if you have nothing else to do at home for the next 2 months, it might not be the worst thing. Yeah.
Might as well UV irradiate all stuff.
Yeah.
And I'm sure in this culture, if you made one, you could sell it, whether it works or not.
Yes.
Yeah. Just wear sunglasses.
All right, we're going to stop there because we're going to come back on Friday and we can grab more of these emails. They're all great. As I said, you can find the whole text of them at microbe.tv/TWiV if you have more questions. TWiV at microbe.tv, and consider supporting us. We do appreciate all the new people who have joined to support us. Thank you so much. It helps with our expenses. And you can go over to microbe.tv, contribute for couple of different ways that you can do this at this special TWiV midweek TWiV. Highly unusual.
I want to point out for those who are contemplating contributing that this is a labor of love. Nobody's making any money off this. Okay, the money goes to buy essential equipment and occasionally help us travel places so that we can spread the word. But sure. my salary on this for the last 10, 12 years has been the satisfaction I get out of doing it.
Yes, well put. We don't get salary, and it's all for expenses, that's for sure. We're not making a killing on the coronavirus epidemic. Many people are doing things that make a lot of money, and it's fine for them. But for us, we want information and knowledge to be freely accessible to everyone. We hope you appreciate it. Joining me today from Austin, Texas, he's an emeritus professor, University of Florida, Gainesville. Rich Condit, thank you very much.
And a grandpa. Sure enough, always a good time.
Ori Lieberman is an MD PhD student here whose rotations have been canceled. You can find him on the last episode of TWiV and also over at This Week in Neuroscience. We're going to do one beginning of April. Thanks, Ori.
No problem. Thanks for having me.
Ori, I enjoyed this. This was great being with you.
Yeah.
Nice to meet you guys.
Alan Dove's at turbidplaque.com. He's on Twitter as Alan Dove. Thank you, Alan, and thanks for arranging some of these emails.
Yeah, thanks a lot.
It's always a pleasure.
It really helped.
I'm Vincent Racaniello. You can find me at virology.blog. I'd like to thank ASV and ASM for their support of TWiV. Ronald Jenkins for the music. This episode of TWiV was recorded, edited, and posted by me, Vincent Racaniello. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.